Aloha. Welcome to the presentation of our ICCV 2025 paper. In this work, we developed a novel contrastive strategy that implicitly disentangles compositional and conformational variances of protein images and enables simultaneous identification of protein compositions and conformations in an unsupervised manner, solving a never-before-solved problem in principle. Proteins or macromolecules can vary in both composition and conformation. Composition refers to changes in the shape components themselves, while conformation refers to changes in how those components are spatially arranged without altering their individual shapes. Our cells are filled with countless proteins, each existing in different compositions and conformations. Understanding these variations is key to uncovering many biological processes working inside the cell. With advanced bioimaging methods like cellular cryo-electron tomography or cryo-ED, we can now visualize proteins of diverse compositions and conformations in their native cellular environment. Cellular cryo-ED provides a large, noisy 3D grayscale volume called tomogram that contains a mixture of numerous 3D protein images called subtomograms. Given this collection of noisy subtomograms, our goal is to identify which protein each one represents. That is, determine both its composition and conformation. We approach this problem from a content transformation disentanglement perspective, where for each image, we obtain two disentangle latent factors. One is the content factor that accounts for the compositional variability. Another is the transformation factor that accounts for the conformational variability. Thus, we reduce the problem to unsupervised content transformation disentanglement. Currently, there are a few solutions to this problem in the natural image domain. These methods perform explicit disentanglement of content and transformations, where a known explicit parametric functional form of the transformation factors are used. However, conformation does not have such a known parametric functional form, making these methods unsuitable for the task. Consequently, we relied on implicit disentanglement, where the model implicitly learns to disentangle a transformation without using any explicit parametric form of it. This sounds a bit like magic. If the model does not explicitly know anything about the transformation, how can it disentangle it? Well, past research has shown that self-supervised learning with data augmentation not only disentangles the transformation used for augmenting, but also other transformations in practice. This opens a new avenue for our research. Can we use a simple geometric data augmentation like rotation and disentangle conformation-like transformations implicitly from the model? Can we do it with existing self-supervised methods, or a new method is required? We indeed find that the existing methods were insufficient in this regard, suggesting the need of a new method. To this end, we propose dual contrast, a novel self-supervised learning strategy that, while using only rotation to perform data augmentation, can implicitly disentangle conformation or conformation-alike transformations from content in an unsupervised manner. Our method performs variational inference of both content and transformation using a variational autoencoder framework. For each batch sample, we create both positive and negative pairs with respect to both content and transformation factors and use standard contrastive laws where positive pairs are pulled together and negative pairs are pushed away in their corresponding representation space. We simply rotate the sample to create positive pair for content and negative pair for transformation. We do a batch permutation to create negative pair for content. To create positive pair for content, we designed a novel strategy. Since it is not possible to perform conformation-like transformation directly to the data space, we depend on the latent space. We obtain two batches of samples by permuting the sample batch, then invert the content factors for each permuted batch sample. We sample a transformation factor from random normal distribution and use it together with an inferred content factor to decode positive pairs with respect to transformation. Here we show an example of our contrastive pair creation strategy with simple MNIST degrees. For further details on the training, please refer to the manuscript. By training a convolutional BE framework with our contrastive training strategy, we could implicitly disentangle viewpoint transformation from the class content in the line mode RGBD image dataset, which the existing explicit methods like Vitae, spatial BE, or harmony could not do. Our quantitative evaluation of expressiveness and disentanglement of the latent factors in the line-mode dataset further validated our claim. Here we also observed that the existing self-supervised learning methods with data augmentation such as SimSailor, SimSiam, Moco, or Barlow twins were unable to provide the desired disentanglement. For details into the evaluation process, metrics, and their calculation, we refer to the original manuscript. Then, we created a synthetic experimental dataset with 4 different compositions of proteins: nucleosome, pre-fission and post-fission spike protein, and fatty acid synthase or FAS. Among them, we used 6 different conformations of nucleosome. We collected the experimentally resolved 3D structures of the proteins from protein data bank. Then we created realistically simulated images of these proteins. In total, we created 18,000 images, each are 3D grayscale volumes of size 32 cube with SNR 0.1. The images contain the structures in diverse orientations and shapes. For further details, we refer to the manuscript. We applied our method and the baselines against the synthetic dataset. We visualized the UMAP of the content factors achieved using each method. We can observe the content factor of dual contrast perfectly clusters the images into 4 distinct groups, each for a particular protein composition. We do not see such separation in other methods. For recovering the structures, we performed Gaussian mixture model clustering on the space followed by reliant refinement to increase the signal and resolution. We could successfully recover all 4 protein compositions using dual contrast, which was not possible with other methods. Then we assessed whether the transformation factor in dual contrast can enable identification of different conformations of proteins. Here are nucleosomes in the synthetic dataset. We observed that none of the existing methods could disentangle conformation through their content or transformation factors, and hence they could not recover the different conformations of nucleosome. However, our method represented the conformation through its disentangled transformation factor, and leveraging it, we could recover 4 out of 6 distinct conformations of nucleosome from the dataset in a completely unsupervised manner. We further quantitatively assessed the unsupervised composition and conformation identification performance. To do this, we used 2 key metrics: the adjacent rank index or ARI, which measures how accurately the latent factors capture distinct compositions or conformations, and the area under the curve of the Fourier shell correlation or AOC-FSC, which reflects the quality of the recovered 3D structures. We found dual contrast to show far superior performance than the baselines, confirming our previous qualitative findings. In summary, we explored a largely unexplored area of implicit disentanglement property of self-supervised learning with data augmentation. Leveraging this property and a proposed novel contrastive learning strategy, we performed completely unsupervised identification of different protein compositions and conformations from their 3D cryogenic image dataset, a problem which was not tackled before. However, we solved this problem in principle through realistic synthetic dataset. Doing the same for real cellular datasets presents additional challenges, such as more extensive upstream analysis to handle higher noise levels, and the need for expert validation in the absence of ground truth. Moreover, the implicit disentanglement achieved through self-supervised methods may have broader applications beyond this setting. We leave these directions for future work. Thank you for watching the presentation. If you have any questions, please email us at the email address. Mahalo.